Hello, everyone, and welcome to today's presentation titled Empowering AIDD, Building a Faster and Smarter Platform for Cancer Drug Discovery. This webinar is a part of the 13th annual event in the Cancer Research and Oncology Virtual Event Series. I'm Melanie Chan of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Yok Hian Chionh, Director of Protein Sciences at GenScript. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Yok Hian, you may now begin your presentation.
Hi, and welcome. Today, I would really like to talk about how we have been building a faster and smarter platform for cancer drug discovery. So here at GenScript, we have been really looking at how AI has been impacting traditional antibody and protein drug design and seeing how we could really improve the process in which antibodies are being generated for both screening purposes and for subsequent in vivo experimental uses. And to that end, we have been iterating, improving our TurboCHO platform so that we are increasing the performance in terms of both expression, amplification, and ultimately also delivery times to our customer. And we do so by really addressing what are the key challenges we have seen across the industry with regards to AI drug design workflows. And before we even come out with the first molecule, producing the first molecule, we really triage the huge plethora of sequences that come to us and look to make the right molecules for our customers. Also optimizing our expression in cells, predicting the best purification path, and subsequently delivering both the molecule and data to our customers, in record times. When we really think about cancer drug therapies, especially in the field of biologics, we had been looking at several FDA approving biologics and looking at how they have evolved over time, right? We have seen a huge increase in bispecific antibodies, and we only see that, I believe this is really the tip of iceberg, more, I believe, as we are able to design more and better molecules, the complexity will increase. But it's important to keep in mind that these are not natural molecules. Evolution does not generate this per se naturally, but rather these are man-made artificial molecules with really no prior knowledge or experience on how to actually make them. And as AI moves from predictive AI in terms of maybe a protein structure from sequence or protein property from sequences to generative AI, in which the language models that's being used to create more and more complex sequences for very specific tasks, right? We are being really hard pressed because the old methods of try and error does not really work. And this is what really we are seeing the transition is occurring, right? Traditional paths, as we've seen 20 years ago, where we inoculate animals, look at the hybridoma, screen for antibodies or specific users, try to characterize them, doing in vivo experiments, and finally humanizing them, right, has been really the workhorse, and we really have huge success on this, especially the cancer therapy fronts. But more recently, we have seen AI crept in, right? More and more AI toolkits are being used every step of the way, replacing what we previously termed, bioinformatics, then big data. And as we see more and more of this creep in, the structure of the molecules, the complexity of the molecules, the design of the molecules have changed fundamentally. What we really see is really on the third panel in this slide, in which a generative AI is currently allowing us to look at these molecules on three main optimization platforms, right? Whether it's structure, whether it is activity or function, and finally developability. So all these three comes together, but one core thing, one core issue still remains, and that is no matter what the AI generates in silico, they must be made in real life and they must be tested in order to generate the leads, right? And this means that the funnel is no longer this narrow triangle, but this is huge flow of molecules and materials that needs to be made and fed back to the system. And this is really where we come in when we think about what the next generation of the CHO platform should be. If we look at CHO molecules, CHO cells, my pardon, beg your pardon, with regards to making antibodies, they are really the traditional and still the current workhorses in this front, right? And very typically, we would just put in two plasmids, a heavy chain plasmid and a light chain plasmid. And, this different cells, as a pool of cells, can have different kinds of expression. Some could make the antibody we want, some could be high producers, low producers, some could make, some things we don't want. And there's this huge heterogeneity within that cell population, and we really need to use screening to look for that one single monoclonal, hence the word monoclonal antibody, right, that we need that to produce this molecule of interest for subsequent investigation. These legacy systems really are built upon linear workflows with almost no feedback loops. We can't go back to say that, look at the other cells, right? And it's very much a black box, with a lot of try and errors and brute force methods being used. What is not shown on this slide is really the idea that purification strategies downstream are also more of an art than a science. It really is dependent on the experience of, say, practitioner, right, that guides a team to really try to purify this molecule out. And so the predictive level is really limited, right? And lastly, of course, with different groups of people, even within companies working on various parts of this, the data sets, whether it's just pure experimental data, computational data, or even metadata, they do not fit. There's no common structured data pool, right, that people work on, and the data lake is lacking, and therefore that means that when we're looking at using machine learning to try to find optimized paths, it becomes a challenge to try to get this standardized. And really, as we see, the in silico part of this, right, evolve and take charge and really lead this new generation of AI drug design, right? Whether you're looking at directed evolutionary paths or using pre-existing structures to look at rational drug design methods, that it always comes in which there needs to be a library to be generated with these protein sequences to look at DNA synthesis and sequences. And all this means that there still is a need to make these molecules using a robust system that can take many, many different kinds of designs and produce them, right? And when people think about this, they don't really even think about maybe even the DNA synthesis, for instance. is one of the issues that we need to solve. And therefore, when we went into this endeavor about 2 years ago, we looked at it and looked at the design parameters that we really want to fit in to this platform that supports AI drug design. That includes format flexibility, right? Whether we are talking like a traditional monoclonal IgG molecule or maybe a VHH nanobody, bispecific, some kind of fusion, it can all be done on the same platform. Whether we are talking about sequence variability, I talked about triaging earlier. So we really want to create an AI tool set that allows us to look at the sequence and really um, with good confidence say, whether we'll face any problems working on this, whether it'll be upstream issue, downstream, maybe it doesn't express well. We want some kind of predictor early on so that our customers don't waste their time and money going down paths that might not be productive down the road. As I mentioned, purification is also an issue, and we really want to come up with a roadmap that standardized purification, right, within the platform. You know, I wouldn't touch more on this on the uniform concentration front, but doing library screens myself previously as well, we note that getting the right concentration across evenly is also very important, right? You don't want to overskill certain data, right? And delivering in the format that the customer can use for their own screening, that's also a huge area. And of course, lastly, as we have constantly seen, the data is actually sometimes even more important than the molecule itself, right? And to that end, we are looking at structured metadata, right? Structured sequence to pull together different kinds of data sets so that we can deliver them almost at real time through API to our customers. And that means a lot of automation and integration, but the information is captured and shared, right? So let's start from the beginning of this platform. And, unfortunately, I wouldn't have time to go through each and every single component. But if you really look at the core of what we were trying to achieve, we are trying to take an antibody sequence design to our customers, run it through, design the correct gene synthesis approach to generate the correct plasmids, choosing the right expression conditions, purifying it in the right way, and then sending it to our customers as soon as possible, without fail. And to the first part, when we do receive the sequences, we really want to look at what people would call developability assessments. To us, is a kind of triaging, right? So that we can flag out problematic sequences that will need more attention on or even a redesign before we even start our work. This really saves everybody time and money down the road. And for this, what we will call our immunoinformatic platform, because at its heart, while we are trying to develop AI tool sets around this, part of the time is really based on bioinformatics, right? There's really different parts to this, and which there's both predictors, structural predictors. There's many good tools out there to AI-based design characteristics, such as how to humanization, or here I wrote kinization, but really from any species to any species, right? And more developability parameters. And as we develop these tools, we actually share them. The GenSmart variable domain sequence analyzer is actually now available online. You can go to our website and check. And we receive, we would like to get feedback. And we have been combining the tools for our own protein AI design tool set in-house. This is the latest build you see down there, in which we ultimately want to have a sequence and with both predictive levels of seeing what kind of properties this will have, what is the best way to express it, what's the best way to purify it, and hopefully we can share it down the road. One example of what we recently built in is the idea of using pre-trained data sets. Right here is a Pro T5, and combining it with random forest regresses to look at a certain developability property, which we believe we will be interested in, especially in the area of formulation. There's viscosity, right? And by building this tool set in, we can then make sure that the molecules coming in can be accessed for a certain kind of parameter that we would care about, in this case viscosity, and that is built in. So as we put more and more tool sets in, we believe it becomes more and more powerful. And ultimately, the idea is to really help both ourselves and our customers look at our sequences from a new light. But it's one thing, to look at, very early predictive levels, but how do we apply this to really real bispecifics or new molecular designs that are coming in? And really, this is meant to address the traditional problem of having our bispecifics. We have very smart designs historically, such as whole and not, very traditional, cross-map, with different electrostatic steerings, right? These designs work, but in a sense, they are also pretty wasteful. There are mismatched sequences, there are impurities that need to be taken out. And many a times the root cause is, both from a biophysical level, right, in which there is a lack of complementarity at different domain interfaces, right, imperfect charge interactions, right, and of course, even on the biological level, right, which has translational inefficiency, causing mispairing of translational rates, and therefore, right, between the heavy and light chain. Therefore, there's, we know chain ratio optimization works, and that's one of the reasons why it does. What our current build of the toolset really seeks to do is to triage this early, look at the structures early, look at promiscuity between heavy and light chain. In this case, in case there is homodimers being formed, right? And also using codon optimization to tweak expression levels, not to make it the highest possible, but really right size expression levels, right? So that heavy and light chains or multi-chains are actually produced in a balanced manner. And here, really, I give through an example from a real case in which we do receive, right? In which, we see a bispecific antibody, traditional whole not design with, electrostatic mutations being placed, right, to make sure that, the heavy and light chain are paired up correctly. But at the same time, when we triage this in our system, right, we realize that, of course, there is the case of mispairing in which the two light chains could be mismatched, right? And so this was flagged out very early on, and it allows our scientists to really take a look at it before the molecule is being made and propose alternative designs, right? In this case, a duet map is being proposed. This design actually involved moving the disulfide bond, creating both steric hindrance and a covalent interaction at another spot, therefore preventing the whole light chain switching as well as homodimerization, right? And there's no before and after, because we went ahead with it. We communicated, our customer went ahead with it, and what this shows, it really produced very good perfect purity results from this design. Expression looks great as well, and this really heartens us because we know that this approach really helped us and also helped our customers get the design they want as soon as possible without the complications involved, right? And this is a great segue into how we are really looking at optimizing, right, even before we get to the protein now, right, the gene sequence itself, right? I think many of you are familiar with GenMap, I hope, right? No, sorry, GeneSmart, my pardon again. GeneSmart is really our flagship codon optimization tool that has been online, free for use for many years, right? Some of you might know that we recently upgraded our GenSmart system. There's now an AI version called GenSmart ZQ that, thus far, as much as possible, when we looked at how we could use codon optimization to improve upon the expression on hard to produce or low expressing proteins, we were generally more successful because we look beyond ribosomal stalling towards looking at the context of cells, context of heavy and light chain as optimization parameters. And as you can see, across almost, currently, almost hundreds of sequences we tested, both in-house, and with our customers' sequences, we've seen generally a big improvement in what expression levels we could achieve. One thing we also optimize for that people don't really think about is we also optimize for how easy a DNA sequence can be synthesized. And this really impacts the timelines our customers eventually feel, because we want to make sure that we can test our constructs in as soon as possible and not get stuck at the gene synthesis phase, right? Another area that we don't really talk about and is not really discussed quite often in literature as well as at conferences like this is really regards to signal peptides and their function, and why we look at signal peptides when we look at optimizing a DNA construct to be placed in a plasmid. A bit of basic biology, right? In a mammalian system, with regards to any nascent polypeptide that is being made into proteins from the ribosome, a single peptide is added, right? This serves as an address tag, but it also, upon its creation, stops translation, means it also plays a role in the expression of that said protein. And why it does that? Because with the single peptide, it causes the complex to be brought towards the ER, right, and translocated within. And within the ER, as many people know, here in the audience would know, that it is really the site in which post-translational modifications are being made, and also the site in which trafficking is being directed into different parts of the cell. So if you want a protein to be expressed outside, extracellularly outside the cell, and with a certain kind of post-translational modification, signal peptides are really something you want to influence and choose correctly. So we developed AI tools at the same time to look at both natural signal peptides that occur in nature, and of course start to develop our own signal peptides with the aim of both increasing expression for otherwise, difficult to express antibody formats, because, as you know, designs could be unnatural by themselves, but also be able to tune those expressions, right? We might want certain kinds of constructs to be higher producers and lower producers, right? And, and here, in this case, SB4 is a naturally found signal peptide that, that in our testing looks very promising. In increasing above a certain default signal peptide is commonly used in the industry. And what we term here GSS PW1 is an artificially created signal peptide. Here we also look at how signal peptides, as I said, are actually removed from the final cell, meaning that if we want to reduce the homogeneity of the product at the N-terminal end, we can use signal peptides to influence this, because ultimately this additional residues, such as the VHS residue you see here, are being created and generated by remnants of the signal peptide. All right, so putting things together, I really don't have time to talk about how we are optimizing cells, vectors, plasmids, media together and to try to optimize you. But the end effect of all this is that we have seen one generation after the other of our CHO platform, starting from 1.0, 2.0, out producing the last one, and we had been increasing the pace in which we're improving that platform. Of course, having the best expression system is not really useful if we can't purify the protein to a level that's usable for subsequent library screens or biological assays, right? And to this end, we really want to generate a roadmap to take out the guesswork with regards to selecting for the right purification path for each type of bispecific or bispecific antibodies or fragment-based antibodies we've seen, right? To this end, we broadly characterize them into various kind of fragment, symmetric, asymmetric antibodies, and look at what kind of impurities would we observe in this. And from those impurities, we would then select the correct kind of columns of resins, rather than letting, what columns or resin is available in the lab, what is in the warehouse, or what is our vendors decide the purification path, right? We really want to make this predictive and instructive, right? So this in our current tool set really works like this, right? In which we get the sequence, we understand the format. Once again, the Smart Viewer Analyzer tool is really online. And this allows us to flag what kind of purification schemes it should follow downstream. And here it actually, we would typically recommend affinity chromatography on the first step, and immediately it selects what is the most best outcome, what is the best purification affinity chromatogram, the chromatography's column and skid that makes sense. If not possible, of course, you'll look at other methods, right? And then you'll select secondary chromatography platforms as required. And how we plan to differentiate it by different biophysical properties of the molecule itself. What this means, there's really now less and less person-to-person variance, right? When we look at how we're going to select our purification method and the conditions they use. And we are constantly optimizing this and feeding in news data, and hopefully this becomes, a fully AI-based approach where, where we can come in and look at molecules quickly and decide, right, the purification path. So here I actually listed two cases of bispecific antibodies, right? They are actually very similar. The one on top, in case one, is really in which we're looking at a heterodimeric antibody with a single VH3 and VH1 difference at the heavy chain, meaning they share a common light chain, right? And to this end, right, we really are exploiting this difference between VH3 and VH1 to select the wide purification path. Right, as you can see, within one step, we can achieve 99% priority, and that is the power of this platform. But for something else that's really very similar, in which there's no common light chain, right? Everything else being the same, right? Then our system actually suggests for us to actually look at salt gradient changes, salt gradient properties using SCEX, right? Because the challenge for this molecule would be for the light chain to be swapped, right? And in which that's step one, and subsequently we will do a gradual decrease in the salt gradient, as you can see here, to actually improve the priority. As you can see, once again, we get achieve 99% priority here, roughly in 2 steps, because we have cut out in a stepwise manner and do a gradient in the second step, right? But this really, I hope, highlights the power of our approach in this, in which we are now creating a systematic roadmap on how we actually approach purification for very varied kinds, the plethora of antibodies or antibody-like molecules out there. And lastly, I would really like to talk about how we have been putting this together and what results we had been delivering on this, right? Some of you might have noticed this science article that came out in February this year, in which, evolutionary scale, right? AI company that's using Meta's ESM models, right? Had the proof of concept of how the AI system, generative AI, could develop over 500 million years of evolution, directed evolution, within a single experiment in which it took GFP, and changed, almost most of the structures out there, but still retain that fluorescent green property. We contributed to this effort by really generating, right, this huge variance of GFP protein variants for these directed evolution experiments in literally 8 business days. Time to see that. But another more recent example, right, maybe one that's more relevant to this audience, is that we were approached to look at produce about 700 VHHFC molecules, and we were given a very short timeline, right? Roughly 6 weeks with pretty strict requirements because this goes into a subsequent screening library. And using our approach, really, we really outperformed. In fact, we had almost, except for one molecule, we actually had 699 of the 700 done within 18 days. And really, how this is done is really during the triage step. We already looked at and flagged out the most important variables to look at, how to optimize gene expression, what purification paths to use, right? And so that it became a question of execution. We did do it in batches, both to de-risk, but also at the same time to allow us to fit into the system. And of course, naturally, they are also batched because, different variants require different synthesis times and scales at the gene level as well as the protein level, and that allows us to put things all together, right? And of course, we include controls for comparison among batches. And lastly, the system also allows to flag out that one molecule, maybe other CROs might say, you know what, we did 699, we are good, right? The one just can't be made. But for us, we took it as a challenge because, coming from a background for drug discovery, the one that got away always irks me. So if we look at it, and we ran it through our system a second time to see what went wrong. In fact, nothing did wrong because we were trying to optimize for as many molecules. But now we can pick out the outliers and look at specific for reasons why it failed and develop specific methods, right? So that we can go back and within 10 days, we generated this. In this case, it was a purification issue rather than an expression issue. So we tweaked the pH for the purification conditions, and we managed to get 98% within one single purification step, right? And meeting all other goals. So all in, I hope this talk gives you a sense of where we are going with regards to platform building, especially to try to support the new generative AI movement with regards to drug design, right? Our method is really, as hopefully I demonstrated, it's really more cost effective for both our customers, right? Because we really want them to make molecules that matter. And of course, it cuts down time because it reduces wastage, right? And whether with regards to expression or purification. And lastly, the idea is to be as consistent as possible, whether you order a protein from our site in the US, in Singapore, in China, you expect to give you the same quality of products anywhere, right? That's all I have. I'm happy to take any questions. Thank you.
Yok Hian, for your informative presentation. We will now begin the live Q&A portion of the webinar. And it looks like we have quite a few questions that came in already. So if you have a question you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. We'll answer as many of your questions as we have time for. Now, let's get started. How do you balance AI-generated antibody diversity with manufacturability constraints, especially for complex formats like bispecifics?
Well, that's actually a pretty good question. Well, you have to note that, at GenScript, we generally leave the design itself to our customers, but it does come down towards the developability angle. For developability, sorry, quite a mouthful. As you can tell from the presentation, we built in individual narrow AI tools every step on the way, from the triage side, to downstream to actually deliver itself. So this means that, even before a single antibody has been made, we actually look through the sequences, and then and through our own internal algorithms, right, regards to, it's literally a multi-parametric optimization, and we look for, whether this is prone to aggregate, whether there's, sequences that, have different charges and so forth. For bispecifics specifically, we're looking at whether there is charge imbalances, whether looking at the geometry makes sense, right? And all this gets computed into a score. Typically, when we look at something, we tend to dump them into low, medium, high bins, and then, using a score-based system, we will say, okay, this, we can possibly make this, versus this design doesn't make sense altogether. And ultimately, so far, we have been pretty successful with this new system. About 75% of problematic sequences get filtered out, and then we make the remaining, and sometimes we learn along the way as well. So, hopefully that answers the question.
Thank you so much for that. The second question that came in asks, what experimental validation is required to trust AI predicted stability or solubility tags, and could over-reliance on in silico tools introduce blind spots?
It's weird because I already answered the question in a sense, but just like what I, just like having to repeat myself, the problem with AI models, as we move from predictive models at this point in time to more generative AI models, as we can see, with the new generation, is the problem of overfitting. The gist of overfitting is, when an AI is exposed, disproportionately to many kinds of, a certain kind of literature or data, it tends to generate response in that manner. And therefore, when we are looking at, designs, and we are looking at the best path to generating a certain antibody, we typically take it within the context of that being a hypothesis to be tested, rather than something that, is supposed to be true. What this means in practice is, when we actually look at certain decisions our own AI tool gives us, right? Like that you make a prediction about solubility, make a prediction about aggregation. It is more of, it is less prescriptive and more of something that we need to test out. And often they're not. No, we see that it gets it wrong, and rather than throw away that result, we actually feed it back into the model, so that the model actually can use it to learn, and so we don't over-index, a more technical term here, on any specific result or data set. Right. Yeah, I guess we can move on to the next question.
Thank you, Yok Hian. And then the last question we did receive actually is also about AI, and it asks, for teams adopting AI platforms, what's the biggest technical hurdle you had seen? Data standardization, computational expertise, or integrating wet-dry lab workflows?
Yeah, I know. I think it's more the human factor behind AI and how we adapt our workflows. For honestly, AI at even at this stage, and even in 5 years' time or 10 years' time, I don't think it will fully, replace our bench scientists. Right. So and that's something that we need to take note of, because what AI is and developed for is actually a tool, and is meant to augment human intuition. So it's supposed to help with tasks that are repetitive or requires a lot of feedback, as well as ideation. Those are what is currently great for, and I think this is being developed further. So yes, standardization, data standardization is a bit of a pain. uh, that's because there's a lot of legacy systems, and I'm sure anybody that work in the wet lab would know that writing your lab records is really not what you are looking forward to do every day, right? So but ultimately, getting that data set in a standardized manner, in a data pool that makes sense, right, and in a structured way, is really what's generating important to generate a good model, right? And the next thing is really to getting our scientists to actually um, a uh, trust the results but verify. I think I covered that in the second question as well. And really, what we do is actually have challenge sets. Sometimes we know it's wrong, we put it in just to see whether whether the AI would lie or something, but also let us to see if any values can be further tweaked, right? And lastly is to prevent the opposite from happening. That means we want to prevent our scientists from over relying on AI to solve questions or problems that it is just isn't meant to do, right? So that's usually how we see it matters, because from understanding very changes in minute shift in different predictions to actually getting our scientists to work with AI in a productive manner. That's actually something we are still doing, and we hopefully could better ourselves. And quite often than not, we found that co-locating teams and building very interactive dashboard with no coding required for biologists usually allow us to work better with AI. And so yeah, you know, it helps us do our science faster, but the AI really doesn't do the science at all.
Thank you for that. And thank you, Yok Hian. Do you have any final comments for our audience today?
No, thank you for joining us today. And if you have any further questions, feel free to drop it to our website and contact me. And we'll welcome everybody to try our TurboCHO service.
Thank you again, Yok Hian Chionh, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall after the webinar for more information and resources. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until October 1st, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.